Good morning, everyone. Thank you for the nice introduction. I'm honored to be here in this meeting from the Ohio State University Extra Medical Center, USA. Like you mentioned, it's currently 12:45 a.m. And I must admit I'm a bit drowsy, but I hope to do a presentation. But I hope you all have been energized by morning coffee. Today's my talk is on, like mentioned, vaccine development. That's the title of my talk is Future Vaccine Strategy, Thinking Outside the Box. So my involvement in this field for vaccines span around 14, 15 years. I started vaccine research when I was at NIH as a fellow. So I got an opportunity to work with pioneers in HIV vaccine field, and I have contributed some into the HIV vaccine development in general. So I was fortunate to work with the first and so far the only HIV clinical trial that has shown some protection from HIV, which called RV144 study. So in 2017, I moved to Ohio State Medical College to establish my own independent research program. And I'm continuing working on HIV vaccine development since 2017. Next 45 minutes, I like to discuss current status of vaccines in general and how we can improve current vaccines and develop new vaccines. Try new ideas to create new vaccines, and what are the challenges that we have for, basically different aspect of the vaccine development. So I have nothing to disclose. So this is a disclosure statement that we have to do in every presentations. We'll begin with the historical foundation. So the story of vaccine development is not one of it's actually a determination and curiosity. If you take the history, so as we know, Edward Jenner and Louis Pasteur, the famous figures that laid the foundation for the progress of the vaccine science. Like we know, Jenner found the smallpox vaccine in 18th century. And this was a big step, showing that control exposed to a weaker form of disease could create immunity. That's the basis. And then Pasteur continued this idea and expand into the other diseases. His work actually basis for many killed vaccines and weakened vaccines that we are still influence our vaccine strategies in our day. So even before Jenner's time, there was a practice called variolation. Actually, in this variolation, it used materials from smallpox to give basic protection. So this shows that this controlled exposure could help make vaccines. So this concept is not novel when the Jenner started this, vaccines, but it was practiced even 3000 BC. So again, so what has Jenner done at that time? He actually bring those things into the real clinical practice and in the science. So I would like to go through some past history. So we have witnessed some deadly diseases transform to a controllable disease or eradicate during the past about 100 years because of the vaccines. You can see in this slide, we have completely eliminated smallpox, diphtheria, polio from United States and other world for past couple of years. I mean, past like 20, 30 years. And also rubella, mumps, pertussis, and measles, they were controlled for a great extent. And actually, they decreased about 9-10%. It's like eradication. So in this slide, you can see there are many challenges in our human history. It's many diseases that challenge us. For example, typhoid fever, meningitis, whooping cough, polio, and so on. So, and it shows that the discovery of the cause of the disease and the approved vaccine. For example, typhoid, the cause of typhoid, which is a Salmonella typhi, was discovered in 1884, but it took a long time to develop a vaccine. The licensed vaccine for typhoid fever, actually, at least FDA approved vaccines came to light in 1989. So it took a long time. But when you go to the chickenpox, measles, hepatitis, so on, and even for the COVID-19, it was last year or 2 years ago, 2 to 3 years ago, it only took a few months to identify the causative agents and come to the develop the vaccine and get the FDA approval. So we can see that how vaccine science developed from late 800 to nowadays, how vaccine strategies develop, and our technique and knowledge and again the resources develop into this level. So again, those vaccines that we have seen up to now, so it's developed based on conventional vaccine strategies. Again, I'm not talking about COVID, but all the other vaccines before were developed based on some of the conventional strategies developed in the past 100 years. So starting from the weak form of microorganism that use the live attenuated vaccines, for example, in measles, mumps, rubella, and then it started using the inactive form of the virus and also killed virus, which is polio and SARS. So there are many techniques used in the past in order to develop the immunity against the infections. Like I mentioned, weak form of microorganism, which is called a live attenuated virus, inactive virus. So the other form is that the purified pieces of microorganism, which is called a recombinant protein. So they can purify it, or you can make it in vitro and then use purified and use. So this is a recombinant vaccine, and many bacterial vectors used in the past. And also there are DNA-based, RNA-based, mRNA-based vaccines came very recently, but viral vectors was used in the past. So again, there are conventional strategies which will help us to develop good vaccines which are protective for many diseases. And the approaches that we have used in order to vaccinate people, population. So it's mostly oral, vaccine delivery methods, oral vaccines, for example, oral polio vaccine, rotavirus, and intramuscular vaccines, subcutaneous and intradermal. Those are the conventional vaccine delivery platforms we have used up to now. So, however, there are certain diseases which we still have challenges. So we don't have a good vaccine developed in order to prevent those diseases. So with all our achievements, so we have a good vectors, we have good vaccine strategies, good delivery routes, but still we have challenges. For example, HIV. It's starting from 80s, still there are lots of research being done to try to, and there are lots of clinical trials being done to get to develop a good HIV vaccine, but still we don't have a HIV vaccine. And HIV pandemic is a major disease in the world. And also take malaria. Malaria still affects millions in the world. We don't have vaccines yet. RSV. So, and TB, of course, we have a TB vaccine, which is a BCG, but it's not a preventive vaccine. Dengue fever, killing thousands of people, but we don't have a good protective vaccine, although we have one vaccine which licensed last year in US, which is called DengueVax, which started last year. and for Ebola, there's a one vaccine developed very recently, but it's also after so many episode of Ebola in West Africa and in different part of the Africa. So again, Zika, we don't have a vaccine. So we are on a mission to find a vaccine for these infection which listed here, and some vaccines, some diseases which can come in the tomorrow. So even though we had some victories in the past, there are lots of works to do in the world of vaccine to understand the vaccination process, the principles, and new strategies in order to combat these diseases. So like I mentioned before, we don't have a vaccine for HIV and for TB and norovirus. Fortunately, we have some vaccines developed for dengue, and I've seen some countries use, and including USA. And RSV vaccine is just approved, I think 2 days ago in the United States for pregnant mothers. But up until then, so people were dying with the kids and elder people were dying with RSV, complication and RSV. In Zika, we don't have a vaccine yet. Malaria vaccine was obvious. There's a vaccine developed like a couple of years ago by Pfizer, and now it's approved by WHO and used in different countries. Still, there are many challenges still persist in the vaccine field, and also when we take the BCG, of course, TB, control TB, so we have a BCG vaccine, but it's not a protective vaccine. So what is the real problem? So those are the things we need to understand when we think about the future vaccines and different strategies we need to use in order to understand the pathogenesis, immunology, and also develop the better vaccine for to combat these diseases. So one example I want to give you here, the challenge with the vaccine development. In the case of HIV, so I think most of you know that HIV epidemic started in 1980s. Right after the pandemic starts, so people thought, okay, this is a viral infection. The notion is that viral infection can be neutralized. The virus can be neutralized by antibodies. So the first clinical trial for HIV vaccine start with developing antibodies by using protein, so protein-based vaccine. So the first trial was conducted in 1987 at NIH by a set of scientists. So this is a phase 1 trial. They did this study before we went to the clinical trial. They did this study in the monkeys. They found that when you use GP160, which is a part of HIV, and injected and when vaccinated with that, there's a good antibodies in the monkey. So they thought that having a good antibodies against the HIV binding protein, which is GP120, can prevent the disease. So they did the clinical trial in 1987. But the results was that having a higher titer of antibody does not prevent the disease. So again, so they did many studies, large scale 2 clinical trials in it's called VAX 003 and VAX 004. Again, Sanofi and Vaccine company, the 2 drugs companies, they developed this vaccine based on protein base and try to see whether they can prevent the HIV. It didn't work. But same vaccine strategy using hepatitis B, which means the protein-based vaccine for hepatitis B works. So it's all depend on the bugs that you are dealing with. Some vaccines work for some bugs, but some vaccines not working for other bugs. So that is very important to understand. So and the second HIV vaccine clinical trial was conducted by Merck actually. So that was a T-cell based vaccine. It's also in 2004, 2007, between that period. So they used the adenovirus vector because of the first trial they failed. which is the protein-based vaccine. And in the adenovirus vector, it's known to mount T-cell response. So they think that having a good CD8 T-cells against the HIV can prevent the HIV. So Merck went for this trial. There are two trials actually they have done. One called a STEP trial and other trial called Kimberley trial. Both trials were happened in the North and South America and also South Africa. So in this trial, what they found was not just these vaccines failed, but also they found that those vaccine recipients who had this Merck vaccine had more chance of getting infection. In other words, those got the vaccine, they had a higher chance of getting infection. So those two vaccine trials failed in early 2000, 2004 and 2007. So it's a big blow for HIV vaccine development. And also that educate us. So using different strategies for vaccine development is necessary, and we need to try different strategies, and we need to understand the molecular mechanism and also immunological mechanism behind the vaccine protection. So what are the barriers to the vaccine development? So that is one thing we need to understand if we want to develop a protective and efficacious vaccine. So there are many factors. I have listed here 5 main factors. So we need to understand how immunity develops. So that's one challenge in the HIV vaccine development in the early days. So we don't know how the immunity to the vaccine to HIV. Still, we don't know. So understand how immunity develops. And then we need to understand host variability. So the person who acquired the disease, we need to understand there are differences. I will come to this in a little details in next few slides, but I just want to give an overview. And then also we need to understand the pathogen variability. What it means, for example, in influenza, we have many variations of influenza. HIV, we have many clades. So we need to understand pathogens and their variability. And also we need to understand the vaccine safety, because it's one of the major concerns. And also the environmental factors, geographic factors. Those factors actually we need to understand very well in order to come up with a good and efficacious vaccine strategy to prevent the diseases that we are going to face. I mean, the diseases that we have right now, which we don't have vaccine, or the diseases we may face in near future. So, so in getting to little details about understand how immunity develops. So again, I'm not going to do an immunology lecture here, but I just want to give some points how the immune system works against the antigens. So you have when you expose to an antigen, so you get the primary response, and then if you expose the same antigen second time, you get the secondary immune response. So always secondary immune responses is high and high steep because it's a memory kind of response. So again, T cells and B cells. So basically, the immune system recognize the antigens and innate immune system recognize the antigen, present the antigens to the adaptive immune system, and they have two different phases of the responses. So basically, I know it's a immunology 101, but we need to understand this mechanism. Obviously, we know that this is the way the immune responses to normal pathogen or any antigen exposure. However, there are lots of things behind this immune response need to understand in order to develop a good and effective vaccine. So one of the major things that we need to understand is the correlation of protection. How does any viral infection can ameliorate? So how does you can fight against any challenging organism. So we need to understand what are the prominent immune responses needed for that to combat this bacterial, virus, or whatever pathogen. So we need to really understand the molecular phenotype and immunosignatures in to the different organisms. So again, one thing is, like I said, correlates of protection. So correlation of protection means what are the important factors which can prevent the biomarkers that associate with the protection from disease. So what we can do that? So for that, actually nowadays there are lots of techniques developed: high dimensional flow cytometry, transcriptomics, single cell RNA sequencing, so and so forth. But at the same time, when we understand those different techniques and also use those, we need to use those techniques in order to understand the interaction, the complex interaction between the pathogens and your immune system. And also another one important thing neglected in the past, vaccines, but we need to, of course, focus on more is innate immunity. Because now innate immunity, we know that previously that innate immunity, it's just a first line of defense. But now we know that it's not just first line of defense. It's also, it has a adaptive immune responses, which called innate memory responses. So again, for example, NK cells, monocytes, they do show the innate memory response. So we need to understand, and we also need to appreciate the innate immunity, their innate memory potential in order to develop a good and protective vaccine. So next, we go to the host variability. I think I don't want to go into details. It shows that. So the host, their genetic factors, especially HLA genes and HLA gene polymorphism, it's always associated with the influence of immune response to viral infection. In the HIV, it very clearly shows that certain HLA type patients have a propensity to resist to the HIV, certain strains. So there are lots of things we need to understand even in the host. So we need to understand the protective biomarkers, population, again, different population. For example, as in the West European community, they some communities, they don't get HIV even they expose, because they have a Delta 32 knockout. They have a polymorphism. So Delta, that means they have a CCR5 mutant form in their genetic makeup. So they don't get HIV. So even they expose. So there are many things we need to understand. So, and also there are lots of variations in the different age group, and in the male and female sex group, and also ethnicity. So some of the some of the populations have more propensity to get the disease than the other in the same organism. So those are the things which we need to understand if we want to develop a good vaccines for whatever the infections which we will get in future, or we will challenge by in the future. So how we can do that? So we need to understand the genomic-wide association studies, or GWAs. So we need to put our full of energy and understand the host variability, and we need to map all these immunodominant epitopes, and also we need to understand any HLA typing or any other factors which are associated with the immunity. Again, I think this is very clear. We know that pathogenic variability, because we know about different virus and different clades and different strains. Even in the COVID, during the COVID, we know that how many variations, variants came after COVID pandemic, after start the COVID pandemic. But we need to start thinking about this pathogen diversity, starting from one organism, one particular clade, and it's evolved with the human, with us. It's changing. So we need to understand. And also antigenic drift and shift. Again, influenza, when we take the influenza, it's changing every year. So we have we know that influenza vaccines or the flu vaccines change every year. FDA give the they predict the whatever the possible the virus circulating every around every August or so in every year, and they make the vaccines to the different variation. So it's there. So the pathogenic variability we need to consider when we are developing the vaccine. It's a complex biology, because it's not something which is simple we can predict, but it's very complex process. And also we need to appreciate the latent infection. Some infections, so they go away, but they are persisting because they are in as a the virus is hidden there as a latent, so they can reactivate. So there are lots of things to understand. So host pathogen interactions, that's another one important things we need to understand when we are thinking about developing a vaccine. Again, there are certain things like immune invasion. So the bugs are really talented. They know how to escape the immune pressure. So we need to understand what are the factors which can develop immune evasion. So how we can do that? There are many other techniques that use developed in the molecular biology and also genomic science, DNA protein interaction, epigenomics, metabolomics, those kind of techniques. And also thinking about broadly neutralizing antibodies. Again, that's one of the major things now we discuss in the HIV vaccine field, because if you have a broadly neutralizing antibodies, we can prevent all these HIV clades and we can prevent HIV vaccine. HIV infection. But can we have a vaccine in order to develop a broadly neutralizing antibody? That's a challenge. None of the clinical trial success up to now to develop a good antigen which can generate broadly neutralizing antibodies. Why? We don't know. So, other barriers to vaccine development, obviously those are not biological, but it's actually social mostly. So adverse events, again, sometimes you get the reactions or those things we need to deal with that. And vaccine hesitance, I think the biggest hurdle in vaccine development is this vaccine hesitance. So I will, if I have time, I will explain one incident that I experienced 2 weeks ago when I came to, I was in Sri Lanka like two weeks ago. So I had experience with this vaccine hesitance in real world. So, and also environment factors, poor nutrition, poor infection, again, poor immunity, because people are malnutrition. Malnutrition certainly affect on the vaccine protection, that's especially happening in the West Africa and African regions. So if you want to develop a good vaccine, what are the strategies we need to use? Okay, so thinking out of the box. So this is the title of our talk today. So we need to understand where we should start for new vaccine development. So in the past, it's true that we had vaccines. It was that vaccines prevent most of the disease, but not anymore. So we need to go more in details, and we need to understand if we want to face this challenge. So the term vaccinomics is really new term. It's developed. Obviously now it's used in the vaccine with the vaccinologists. So the term vaccinomics means using genomics tools, which I'm listed before, single cell RNA sequencing, RNA protein interaction, proteomics, and so forth, and use those the tools in order to understand the complex biology between host and pathogen. And those things we cannot do just by using general tools. So we need to use those tools in with the database and analytical approach. So we are using bioinformatics together with genomics tools in order to picture it, in order to model the vaccine effectiveness or host pathogen interaction. So in that host pathogen interaction, we actually can predict what are the new vaccines that suitable for given disease. For example, like I mentioned, so protein vaccine is not good for HIV, but it's good for hepatitis B. So likewise, we can use the genomic tools by using the bioinformatics analysis, and then we can predict the different vaccines strategies, vectors, different vectors which we can use for develop the vaccines. Do we have to use a passive antibody as a therapy? That's what's used in the HIV field. Broad-neutralizing antibodies, where recently they are success in by using broad-neutralizing antibodies. So, and also the one other point which I forgot to mention about the adjuvants. So adjuvants are the substance which mimic some innate immune response and which mimic pathogens and trigger innate immune response. So we can predict those novel adjuvants by using this approach. And why we need to do all these things? Because we want to combat these pathogens which come in tomorrow, or whatever the vaccine or whatever the organism that we have right now, but we don't have good protective vaccines. So by using those technique ideas, so scientists came up with different vaccine approaches, innovative ideas. So I will list some of this here. I think I don't have much time to go over everything, but I will list some of the important things that are used in nowadays. So for example, HPV, human papillomavirus vaccines used now, and it was developed at NIH. What they have done was they use actually virus-like particles. Actually, initially it was a computational modeling, and then they came up with these virus-like particles which mimic the they don't have the genetic materials of HPV, but they mimic the virus, and they can provoke robust immune response. So virus-like particles used in the HPV vaccines, and also there are many other experimental vaccines they are using the VPL or the virus-like particles. And DNA virus, I think now we know mRNA vaccines because it's used heavily, used in the Moderna and Pfizer BioNTech vaccine, but DNA vaccine was developed just before that, actually. So DNA vaccines, it's very it's tested in HIV vaccine fields. Actually, some of my I have some personal experience with how it's testing in animal models and in phase 1 clinical trials in humans. So DNA vaccines, it's DNA fragments which it can it's represent the produce the viral protein which can elicit immune response. There are many advantages by using the DNA vaccines because it can target the CD8 T cells. You can get the robust CD8 T cell responses, and also the TFH, which the T follicular helper cell responses, which needed for good B cell responses. So there are another approach, nano particle vaccines. Again, nanoparticle vaccines were tried in SARS-CoV-2 because in that case, so you can have a different type of proteins like from the pathogen, and they can those antigens can incorporate in the nanoparticles and make as a vaccine. So again, the nanoparticle vaccines, there are lots of the advantages because you can so in the vaccination, so what we are expecting is to send the vaccine antigens to the right place where it's a delivery method. So there are many methods developed with the nanoparticle vaccine to send, for example, mucosal to boost the mucosal immunity. So some nanoparticles make in such a way to deliver into the gut or the GIT tract. So there are many things manipulating in the, we can manipulate the vaccine delivery platform by using nanoparticles. And also we can, not just one particular protein, we can deliver many particles by using nanoparticle vaccines. Again, that's important because previously, in previous vaccines, so we had one particular protein, we produce that protein and inject or vaccinated and develop antibody against particular vaccine. But now we not just one, we can have multiple different epitopes. We can tie with the nanoparticles and deliver into the individuals, which can boost immune responses. So again, I don't think I need to go in details about the mRNA vaccines. We know the success of mRNA vaccines. So Pfizer, BioNTech, and Moderna used that vaccine. Again, that vaccine was tested even before. So some people think that mRNA vaccine just came after COVID, but that's not true. It was tested many in animals models. So they were trying to use that mRNA technology to have a pan-influenza vaccine. So the advantage that the messenger RNA, that's the genetic makeup, gives the information to produce the protein. So again, it's a very specific, and there's no such a off-target effects. It's just producing the appropriate protein that we needed from pathogenic proteins in human host. So, and then we can elicit the antibodies. So that was, again, I'm not going to talk about that, but we can see the success of mRNA vaccines at least past 3 years. So based on the mRNA vaccines, so there are another type of mRNA vaccines developed is called self-amplifying RNA vaccines. Again, that's an extension of mRNA vaccines. So now they have developed a synthetic part of the, they added a part to self-replicate mRNA. So in that process, what they have done, so instead of using mRNA vaccines produce one or protein or a couple of proteins, but by using the self-replicating tag, they can have they can amplify this mRNA in a great magnitude, and they can get a robust response. Actually, these are not in clinical trials, but they are under investigations under animal studies, but they have shown a very remarkable results. And also these self-amplifying mRNA vaccines shows that they have their vaccine immune response are durable and also robust in many studies, in animal studies, and they are planning to some phase 1 clinical trial very soon. The other approaches are the multivalent vaccines and universal vaccines. Both vaccines are going similar mechanism. So multivalent vaccines, we know that influenza and COVID-19, we know that it's a respiratory disease. So we can put those two bugs, antigens in our same vaccine, and they can deliver to a person. So we can have immunity for both COVID and influenza. So people are trying that. It's like by using different technology, like I mentioned, the nanotechnology and also other methods. So they are trying multivalent vaccines. I think we need multivalent vaccines, not just for different bugs, even for same bugs, for especially for the influenza, because there are different strains of influenza. So they are trying. And also universal vaccine. I think universal vaccine is it's a concept, still it's a concept, but people are trying to use many variants and putting together and use as a universal vaccine, especially for the influenza program. Again, so there are another different methods, different vaccine strategies, vaccine using immune cells, certain immune cells, for example, NK cells and dendritic cells. They are using those NK cells and DCs as a vaccine platform. Especially, I didn't talk about the cancer vaccine here, but in the cancer vaccines, more frequently used NK cell-based and DC-based vaccines. Again, the previous session, it was lots of discussion about microbiome. Actually, microbiome can be used as the vaccine platform. I was involved in one of the study in the rhesus macaque. So we changed the intestinal microbiome actually lactobacillus, and we put the HIV vaccines, and we colonize the monkeys, female monkeys' vagina with these lactobacillus and see whether we can have a use the microbiome in order to use as a vaccine, which we have a nice protection and nice results. Of course, it will have to go through several steps before we go for clinical trials, but it's been tested. So microbiome-based vaccines is another strategy that we can use in order to, when we are having this type of challenge right now. So again, I mentioned about previously the vaccine delivery platforms. So conventional platforms, again, intramuscular, oral, subcutaneous methods. But now we need to understand these bugs that we are dealing with. It's not just accessible by the previous routes that I mentioned. Some, for example, mucosal pathogens. Again, so we need to approach those tissues by different methods. So there are innovative approaches developed to use mucosal delivery, mucosal vaccines. So there are many vaccines developed just to increase the mucosal immunity. So again, like I mentioned about before, the vaginal microbiome vaccines. So again, it's to increase the vaginal mucosal immunity. So there are many things have been tried, and also we can use the adjuvants to drive the immune response to the mucosal tissues. I don't know whether I will have time, but if I have time, I will show you one slide that some of the study that we did for the mucosal, how we can use the mucosal adjuvants to increase the resistance to HIV in the vaginal tissues and in the gut intestinal immune system. So there are many studies going on in this aspect as well. So the personalized vaccine, this is the next thing we need to understand. So again, like I mentioned before, genomics and personal medicine, it's led to understand we need to have a personalized vaccine. So again, it's a challenging process, but it is right now it's using in the cancer vaccine. So basically, in that case, so we need to understand the personal genetics and their mutations, how they respond to the drugs, how they respond to vaccines. So it's also going heavily in research in infectious disease, but it's also used in the cancer vaccine strategies. So all these things I mentioned up to now, it's just different aspect of vaccine development strategies that we can use. Again, but we need to have all these techniques, and we need to think in globally. So if we want to come up with a vaccine for a disease that is spreading throughout the world, so we need to have a global vaccine responses. And also nowadays we are discussing about in silico vaccines, but it means when we have a pathogen in a different demographic, so we need to get those information. We need to pump all these sequence data, their immune response data, everything into a one major hub, and we need to model those things, and that's the only way we can come up with a effective and more reliable vaccine in future to develop. So in silico vaccines, which means we are using all the information which we can get, clinical information, biological, virological, immunological information, and then we will create a network, create a communication between all this information using the AI and also artificial intelligence, and also different algorithms in order to understand the bugs as well as their protective mechanisms. So those are things are now it's heavily used in during the COVID pandemic. So many people use that. That's a global response for the for the COVID, which is lay a ground for many collaborations and many think tanks. And this in silico vaccines is the one of the next generation thing that people are discussing right now. Like I mentioned, I think I mentioned about this artificial intelligence. That's one of the major things. So we have trillions or zillions data right now, even for COVID. So we cannot, the human cannot process those information. So we need to use the artificial intelligence to use the vaccine development, which is happening right now. I know NIH has spent lots of money in order to get all those database and develop those the algorithms in order to understand pathogens and also protective mechanism, and also coming up with the protective vaccines or protective immune mechanisms. So, like I said, all those things I described in past 30-40 minutes, it's the vaccines, how we can come up with the vaccine strategies in order to combat the disease. Right now, we don't have vaccines, or the disease that we are getting, tomorrow or next year. So again, those are the techniques. But the major important thing is we need to understand education and advocacy. That's important. So it's not just vaccine techniques, but we also thinking about vaccine hesitance. Like I said, even the most educated people, they are still have a doubt about vaccine. So I think when you talk to your friends, even your senior colleagues, like I said, I had an experience when I visited Sri Lanka. I talked to one of the senior clinician. I don't know if he or she is in the audience now, but he was very very particular about mRNA vaccines. Was discussing with me for 45 minutes actually, and try to convince me that the mRNA vaccine is a failure and it's not a protective vaccine. Again, so this is the world we are living. So we need to educate the people how we can prevent the disease by having a vaccine. So this is the greatest challenge. I think even the technique we can develop, science we can develop, but also but the problem with the personal attitudes, I think that's be a major challenge in near future for any given vaccine. So we need to be prepared for that. So I think next I think I have another 5 or 10 minutes. So I think so basically I have given the overview of the new vaccines and what are the challenges and how we can develop. But I will go through very quickly how the journey in the HIV vaccine development, because it gives you some sense how the challenges we face during the vaccine development. I think I mentioned these two trials before. Having the developing first antibody-based vaccine for HIV didn't work out, and it failed, and also T-cell based vaccines failed. And not only fail, it increased the propensity to get the infection, which means it enhanced the disease. So based on this, people didn't give up the studies. They didn't give up the clinical trials. They were trying. So the next study they have done, actually, some of my my previous mentor actually involved in this study. So I was also involved in this study when I was at NIH. So in this strategy, so although those two, the strategy failed, the T-cell based and B-cell based, or the protein based and adenovirus vector based, what they have tried here, they use prime boost method, used protein boost, vector prime and protein boost. So this study was done in 2003 to 2009 in Thailand, used the Alvac vector. Alvac is a canary pox bird flu virus, which cloned with HIV genes. And that vaccine shows some efficacy. This is the first ever HIV vaccine to show some efficacy in clinical trial. The vaccine efficacy was 60%. They used unconventional method. They use a prime boost strategy, which was unconventional at that time. Now it's used in a different strategy, but at 2000, I mean, in the time of 2003, it was unconventional. So they used the unconventional method in order to get some protection. The protection was declined up to 30% after 5 years. So this is not a prime time to go for a vaccine clinical trial. But this is the first ever vaccine to show some efficacy. They used unconventional method in order to get this, results. Again, in that study, there were lots of clinical trials done. So based on that, lots of studies done, sorry, not clinical trials, but lots of things done. So they identified immune correlates, HIV possible immune correlates from blood, because this is a human study. We didn't get the samples. You know that HIV is not it's a blood-borne pathogen, but it's an enteric organism. It's a mucosal organism, basically, because they are replicating in the gut. So we need to see the not just blood-associated immune correlates, but also mucosal immune correlate. Can we do it in human? We cannot, because we cannot get the ethical clearance to do the gut biopsies or so on. But anyhow, with the RV144 study, which is the only successful HIV vaccine up to now, has shown that antibodies, particular antibodies, which called V1, V2 antibody against GP120, can protect the vaccine. Vaccine can that can protect from HIV. So some antibodies increase the risk of HIV. So, for example, IgG antibodies in the blood is bad. Actually, they can increase. So they were found some immune correlates. And actually, we were also interested in this more in depth, like I mentioned at the very beginning, involved in the many studies. So we did monkey studies. We went to deep down to mucosal tissues, and we found some interesting results from that. So we found that there are mucosal immune cells also important for the vaccine protection. It's not just in the blood, even in the mucosa, we really have to look into that. So those are the things that we didn't explore before, now we are doing. Actually, in my lab here at OSU, we are trying to find out how we can increase the HIV vaccine efficacy by targeting the mucosal tissues. Obviously, we have some promising data. So this is our vaccine platform, but at the same time, there are many people tried many vaccine platforms. One is called HVTN702. It started in 2016. They use a different vaccine strategy. These studies are multi-million-dollar studies. This study showed promising results in the monkeys, but it failed in 2020. Study was failed. And the HIV vaccine, this HVTN trial was failed in 2020. It's a big blow to the HIV vaccine. And there was another vaccine trial started in 2017. This is the adenovirus-based vaccine. So actually, the Johnson & Johnson COVID vaccine developed in the exact same platform from the same group of people from Harvard. So they developed a COVID vaccine based on this platform. It's successful, but same platform used for this HTV and 705 trial. Actually, that study started in 2017. Results came in NHP. The non-human primate results were fantastic, but the results came in this study in 2021 failed. And only successful results came for HIV vaccine trial was the broadly neutralizing antibody. Actually, broadly neutralizing antibody is not a vaccine per se. It's a passive antibody study. Broadly neutralizing antibody means the epitopes can cover, or the antibodies can neutralize many strains. Again, I don't have much time to go into details because I have only 4 minutes left. So basically, that broadly neutralizing antibodies, when it's given in the infusion, they can show that this antibody is protective for lots of clades of HIV virus. But the challenge is that there's no immunogen up to now or developed up to now to elicit the broadly neutralizing antibodies. So that's a challenge. So now in the HIV vaccine field, people are trying different, like I said, adjuvants. Try to use the adjuvants to elicit mucosal antibody response, mucosal innate immune cell responses. And also another group, different groups try to develop a different technique in order to elicit antibodies or the B cell lineage, which can produce broadly neutralizing antibodies. So there are lots of new technology using the germline targeted antigens in order to develop different protein neutralizing antibody. Unfortunately, I don't have much time, so I will. So again, I want to wrap up here with one remarks about the Moderna mRNA vaccine. So there are clinical trials going on for Moderna mRNA HIV vaccine. So these vaccines results will come very soon, and people are excited about that. But so antibody response is good, but this is a phase 1 trial. But again, we with the experience in the past, I don't know whether this is going to be work out, but hopefully if this mRNA vaccine worked in for HIV, so I think that's maybe a good news for the people who are suffering, and people this is a huge epidemic in the in Africa and Asia. So with that, I think I would like to acknowledge all the people which I worked with when I was at NIH. Still, we are keeping the collaboration, and also my team who's working in my at Ohio State University. So they are basically working with the HIV vaccine development and also pediatric HIV. And finally, I would like to thank everyone for listening. And I think I don't have much. I don't think this is a preliminary session. I don't have a time for answer any questions. But if you need any clarification or any sort of discussion, so I'm available through email. So you can just follow us. Thank you.